Hello, I'm Colleen Croniger, and today we're going to talk about lipid metabolism and the mobilization of your lipid stores and the synthesis of ketone bodies. During the fasted state, adrenaline will be elevated, there will be low levels of insulin, and all of this promotes the phosphorylation of the hormone-sensitive lipase. The stored form of energy in your adipose tissue is your triglycerides. The hormone-sensitive lipase goes through what's called lipolysis, and that means you're cleaving the triglyceride to glycerol and free fatty acids. The glycerol will go off to the liver and be used for gluconeogenesis. The fatty acids will enter into the mitochondria of the liver, and it will be processed and metabolized to the end product of acetyl-CoA. So the fatty acid is transported from the adipose tissue to the liver cell, being bound to albumin. Once it enters into the liver cell, it needs to be what we call activated, and that's this process here. We need to put an acyl-CoA group onto the fatty acids. To do this, use the enzyme fatty acyl-CoA synthetase. This adds that CoA, and now the next step of the carnitine shuttle is possible. The acyl-CoA will transfer the acyl moiety to carnitine to generate acyl-carnitine. The enzyme responsible for this is embedded on the outside of the mitochondrial membrane, and that's part of the carnitine shuttle. You can see it here, it's represented as CAT, but you can also see in books as CPT, which is carnitine palmitoyl transferase. Acyl-carnitine, once it's formed, will go through the mitochondrial membrane using the enzyme, the translocase. Once acyl-carnitine is inside the mitochondrial matrix, the second enzyme, CAT2 or CPT2, will take that acyl moiety off of the carnitine and put it back on CoA. So now we took the fatty acyl-CoA from the cytosol and got it into the mitochondria through the carnitine shuttle. The end product is going to be acetyl-CoA from beta oxidation. Beta oxidation will cleave two carbon molecules at a time. The end product will be acetyl-CoA. Acetyl-CoA is a substrate for ketone bodies. Acetoacetate, and here we have 3-hydroxybutyrate or beta-hydroxybutyrate. The beta-hydroxybutyrate is a stable form of ketone bodies. Ketone bodies are synthesized in the liver, and they're utilized in the other tissues, the heart, brain, and muscle. The reason why the liver can't metabolize it is because the key enzyme for breaking down those ketone bodies is not present in the liver. Basically, why ketone bodies form is that you have a huge abundance of acetyl-CoA. You also have a huge abundance of NADH as a byproduct of the fatty acid oxidation. These together generate the beta-hydroxybutyrate and acetoacetate. Acetone is a byproduct of the ketone bodies, and it is generated when there's a huge amount in the blood, like during diabetic ketoacidosis. You will respire off the acetone, and it'll smell like fingernail polish remover. Fatty acid oxidation and fatty acid synthesis are linked in the sense that you have regulation at that carnitine shuttle. So where I'm drawing that circle is where the carnitine shuttle is. This carnitine shuttle is inhibited by malonyl-CoA. Malonyl-CoA is present during fatty acid synthesis. So when fatty acid synthesis is going on, you'll increase your malonyl-CoA and you'll inhibit fatty acid oxidation. So the two pathways are linked.
Are you pausing it?